Two orders. We're still on track.
Well, good morning, everybody. Happy Friday. The end of the weekend coming up. I hope we're all looking forward to the weekend. I certainly am. Tonight I get to go to take my wife out to dinner to my favorite Italian restaurant, which is here in Newburgh. Rose Marina is what it's called. Best Italian food I've ever had in my life. And I get to go tonight. God, foggy glasses. Well, first things, amazing job, everybody, yesterday. I got to tell you, I've been doing this a long time. And as I told you before, the surgeries generally go without incident. Rats do fine, no big deal. But I thought they were exceptionally good yesterday. I just look at the cleanliness of the incisions. Most of you were able to minimize the size of the incisions that you had to make while still providing yourself a good working area. And you were able to move through it pretty quickly too, which I was impressed. So you should all give yourselves a pat on the back. So anyway, in fact, probably the biggest faux pas yesterday was mine, telling one group to overdose a rat. By the way, did you look at that rat?
This morning.
Is it? Cool. So I avoided killing something yesterday, so that's a good thing. Anything else I need to? So just a reminder that rat care is all on you guys now. So you need to make sure your rats are fed and watered regularly. With 3 groups of 5 and 1 group of 4, there should be plenty of labor to make sure that happens. Sadly, that includes weekends as well. You can't just ignore them on weekends, and so kind of take turns doing that. It is also going to be pretty important that everybody really kind of nail down your experimental protocols with me so you can get started on that. You have a few days because the surgery rats really need a few days to recover. But I expect that Audrey and Tiffany will have the potassium perchlorate made up for you today so that you can start giving your perchlorate rat perchlorate. And basically what you're going to do is you're just going to dump the water out in a water bottle and fill their water bottle up full of that. And then kind of keep an eye on them for a while because they probably will struggle to get into the habit of drinking it because it doesn't taste good to them. But they'll get thirsty and they eventually will start drinking it. Not a big concern if the amount they consume is lower than everybody else, as long as they're getting fluid into them. That's perfectly. And then I will want you to start tracking mass pretty quickly, but again, I'll need you to go over a protocol for that with me. And the reason why we need to track mass pretty quickly is because mass is really the best indicator of animal health. So if you get a rat that starts just declining in mass, that should be a red flag. And that doesn't mean that having a couple of measurements where mass goes down is a big deal. But I mean, if you've gone, you know, a couple of weeks and your rat is steadily declining in mass, then that's something that we take a look at. Okay. All right. And by the way, if anybody wants to, if a group can't all meet with me and wants to sit down and just talk about a mass measuring protocol, that's totally fine, because it's a pretty simplistic thing. There's just a couple of things you need to pay attention to. I'd like to see if you're going to think about this before I tell you to fail. Okay. Any questions about that? Some of you have sent me emails requesting meetings, and I will respond to those after class today. I was too tired when I got home last night to deal with emails, so and I had to cook dinner for my wife when I got home last night, so because she works even later at night. Any questions? Well, again, you guys did an amazing job yesterday. Really proud of you. So pat yourselves on the back. Okay, any non-lab related questions before we get started this morning? All right, and let's get cracking. I think I'm gonna throw in the towel and just go without glasses today. All righty, so last time we finished up, we were talking about competitive enzyme inhibition. Somebody refresh our memory what it means for an enzyme to be competitively inhibited. Go ahead.
It finds the active site and then its form.
That's correct. And how can you use a Lineweaver-Burke plot to identify a case of competitive inhibition? Somebody else. Jack, what's the answer to my question? You can look at your notes.
Pardon?
You can look at your notes.
Can you repeat the question again, please?
I can. How can you use a Lineweaver-Burke plot to identify a case of competitive inhibition of an enzyme?
Do not remember.
You do not remember. Any of the other gentlemen back there want to help him out? I'm picking on the boys.
When KM low
When KM what?
is low,
Okay, is
the concentration is low, or it can be a
okay. So if I use a Lineweaver-Burke plot and plot a curve with different concentrations of enzyme, what will change and what will not change? Okay. The Km changes, but the Vmax doesn't. Why not?
The Vmax is a fixed constant anyway, right?
It's a what?
It's fixed anyway, right? It's just the
And
max.
what is fixing Vmax? So before we even talked about enzyme inhibition last time, we talked about two ways to change the rate of a reaction, enzymatic reaction, or the Vmax, I should say. What are those two ways? Go ahead.
The enzyme concentration.
Okay, that's one way. What's the other way? The substrate. Those are really the only two variables that are involved in an enzymatic reaction. You can change either of them. So in competitive inhibition, we are sort of mimicking changing which enzyme or substrate. I know, it really sucks that I gotta do this at 8 o'clock in the morning.
Wouldn't it be the enzyme because it takes up the enzyme?
Can you speak a little louder?
Wouldn't it be the enzyme because the inhibition takes up the enzyme, it like blocks off?
Okay, all right. So let me paraphrase for you. Because we have an inhibitor that is inhibiting the enzyme in this case, we are changing the, well, we're actually not changing the total amount of enzyme at all in this case, even though we're inhibiting it. Okay, how can we overcome competitive inhibition? Go ahead.
You increase the amount of substrate.
Yeah, you increase the amount of substrate. You basically outcompete the inhibitor. So why doesn't Vmax change? So we're plotting this as a function of increasing substrate concentration. Why doesn't Vmax change?
Because you're at a point where it's fully saturated with substrates.
Okay, so eventually if you add enough substrate to the system, you'll just totally outcompete the inhibitor. The amount of enzyme is fixed, and so you're always going to reach Vmax eventually. Why does Km change? This gets back to the original question. What are we mimicking here with competitive inhibition? Yeah.
With inhibition, it mimics lower affinity.
Yeah, it mimics lower affinity because we have the inhibitor competing with the substrate for that active site. You need more substrate in order to reach Km, 1/2 Vmax. So that mimics a lower enzyme affinity. Does that make sense? What a great example. Yes.
Did we go over the 2 slides before this one?
Oh, actually, I don't have the 2 slides because I copied these into topic 3. I believe so. But if I didn't, it's not a big deal. They were just review slides. Yeah.
I'm so sorry, but so I think I'm having a hard time wrapping my brain around. So when we add more substrate, it's mimicking lower enzyme affinity.
Lower enzyme affinity for the substrate.
Because we're needing to add more.
Yeah. So if I have no enzyme inhibitor, okay, then the enzyme is going to have some specific affinity for that substrate. When I add inhibitor, okay, some of those enzymes are binding to inhibitor, which cannot produce product, and some are binding to substrate. Okay, the inhibitor and the substrate are competing for the binding site on the enzyme. Because of that, when you have inhibitor in the mix, there's a lower number of substrate molecules binding to the overall concentration of enzymes. And so that mimics lower enzyme affinity because it's going to take more enzyme to get to saturation. Does that make sense? So that means Km is going to be, the actual Km is going to be higher. Is everybody following that? Have we re-grasped competitive inhibition this morning? Before I move on and talk about non-competitive inhibition. Okay, so I was asked about the previous 2 slides, and there's one that I will talk about. I'm sorry I don't have the slide here because your silly professor made a mistake in perhaps what slide he stopped on last time. Usually I'm pretty good about that, and when I don't think I'm pretty good about that, I just go back and look at the video, but I didn't do that today. So one slide was just a pictorial example of competitive inhibition, so that's not a huge big deal, but I did have one slide that talked about an example. And that example was, I believe, sulfa drugs, since you're probably looking at it. Sulfa drugs. And sulfa drugs are used by bacteria to make folic acid. Well, actually, I should say the compounds, the sulfa drugs prevent the formation of folic acid, which bacteria use to make cell walls. And so it prevents cell wall formation. It prevents cell wall formation in bacteria. That's how sulfa drugs work. Okay, and this is just an example of competitive inhibition. And so that's what that was. So you can go and take a look at the slide and read about it. But that was the main reason I showed that slide, just to give you an example, a real life example of competitive inhibition. And some of you have pictures of it there. Okay, are we ready to move on to non-competitive inhibition? All righty. Non-competitive inhibitors. Now they are different than competitive inhibitors because they don't interact with the active site of the enzyme. Instead, they are going to bind to some place outside the active site to inhibit the enzyme. Okay, so they will bind to someplace else on the enzyme and they will cause some sort of a conformational change in that active site, or a slight change to it so that the enzyme becomes inactive. It does not prevent, the change is not sufficient to prevent the substrate from binding to the enzyme, however. What it does do is it makes the enzyme substrate / inhibitor complex inactive. So it does not, it cannot produce product. All right? So I am gonna blow by the next bullet point and go to the next slide because I want to give you I want you to think about this a little bit. I'll go back to that if we need to. All right. Here is the Lineweaver-Burke plot for non-competitive inhibition. What's changing and what's not changing here? And we're talking specifically about Km and Vmax. Go ahead.
Km stays the same, but Vmax changes?
Yeah, so we have the opposite of what we saw with competitive inhibition. Km, remember, Km, you can measure that by the x-intercept here, is the same in all cases, but Vmax changes. Anybody got an explanation for this? Well, let's take them one at a time. So why do you suspect, and it's okay to kind of take a guess if you want, why would you suspect that Km does not change in this case? Yeah.
I would assume, well, this is a guess.
That's
I would
okay.
guess that Km doesn't change because the substrate is still binding to the enzyme.
Yeah, so there's no interference whatsoever with binding of the substrate to the enzyme, okay? And so this is why Km stays constant. But then why does Vmax change? Yeah.
Because the inhibitor is increasing.
Okay, so the inhibitor increases, but what's that doing? Well, it's not impacting enzyme affinity at all because Km doesn't change. What is causing, think about it, what is causing a reduction in Vmax as enzyme concentration increases? Remember, there's only two things that can change. Actually, there's only one thing that can change Vmax. There's only one variable. What variable is that? Go ahead. Yeah, the amount of enzyme is the only thing that can change Vmax. So as enzyme inhibitor is increasing, what is that effectively doing? Yeah, it's decreasing the amount of enzyme available to drive the reaction. So what an increase in the concentration of a non-competitive inhibitor does is it mimics having a lower enzyme concentration. So you get a lower Vmax. Okay, does that make sense? Okay, so there's just the text that says that. So the enzyme inhibitor effectively reduces enzyme concentration, whereas in competitive inhibition, the enzyme effectively changes substrate concentration, or substrate affinity is a better descriptor for it. So they accomplish inhibition in different ways, and the line lever Burke plot is a really good tool to figure out which of these two ways that the inhibitor is working. Questions about this slide? Does this all make sense? All right, here's a fun example of non-competitive inhibition. So you all, cut an apple in half, set it on a counter, and it turns brown, right? That actually is driven by an enzymatic reaction. And the enzyme is orthodiphenyl oxidase, and it catalyzes the oxidation of phenols in the apple and causes them to turn dark. And just as a side note, there's actually a very similar reaction that involves the enzyme tyrosinase that converts tyrosine to melanin. So, it's another thing that creates a color change. But what happens is, like, they used to do this in grocery stores. I don't know if they still do this, because this is a little dated. They could spray apples with a substance called phenylthiourea, and this would bind to a copper ion on the enzyme. That copper ion would be a what? I know, it's been 2 days. What would the copper ion be to this enzyme? Go ahead. It would be a cofactor, okay? It would be one of those metal ion cofactors. It would be one of those loosely binding metal ions. Well, this substance interferes with the functionality of that cofactor, and it turns the enzyme off. So it functions as a non-competitive inhibitor. Okay, here's one that I always mess up. So I am going to do my darndest today to get this straight. This is a relatively new category of inhibitor that just showed up in the textbooks, I would say, about 10 years ago. And it's called uncompetitive inhibition. Now, this type of inhibition has some similarities to non-competitive inhibition. The exception is that the inhibitor does not bind to the enzyme until after the substrate binds to the enzyme. In non-competitive inhibition, the order in which the substrate inhibitor bind to the enzyme does not matter. The substrate can bind first, the inhibitor can bind first. But in this particular case, the inhibitor will not bind to the enzyme until after the substrate does. And this gives it a unique quality compared to non-competitive inhibition. So here are the Lineweaver-Burke plots for uncompetitive inhibition. So somebody tell me what you see here with regard to Km and Vmax. So as inhibitor concentration increases, what happens to Km? I know, looking at these inverse axes drives you kind of bats up, doesn't it? Yeah.
When the inhibitor increases, KM is it's the top line, right? It's the top.
Km is down here.
Right, but like the when.
Yeah, this is the highest enzyme concentration. Yeah. So what's happening to Km? Is it increasing or decreasing? Huh? Say the word. That's correct. It's decreasing, okay? Does that make sense? Because a decreasing Km says what with regard to enzyme affinity? Enzyme affinity for the substrate. What does that imply? High or low affinity? It applies high and high. Does that make sense? But it's true. Somebody want to tell me why it makes sense? It actually does, if you think about it. Remember, the inhibitor does not bind to the enzyme until after the substrate does. So what do you think is happening here? Oh, somebody be brave. What's the worst that can happen? You'd be correct. And that's okay. You guys got a long way to go before you're incorrect as many times as I have been.
There's like nothing inhibiting it, so that's why the affinity is high.
When you say nothing is inhibiting it, I'm not clear what you mean by that.
Because the substrate binds first. And then, like, did it say that when the inhibitor binds, does that make it inactive or?
Does it make it what?
Uh,
It does make it inactive. It makes the enzyme substrate inhibitor complex inactive. Yes. But does that influence Km? Well, so we don't get bogged down, I will tell you. What's going on here is the formation of that ESI complex kind of locks all that together. Thus, it enhances the ability and speed with which these enzyme inhibitor or enzyme substrate inhibitor complexes form. So this creates a scenario that when you have a lot of enzyme, it actually kind of ups the affinity, or at least the impression that the affinity for the the enzyme for the substrate is going to increase. In all likelihood, because it is preventing or making it less likely that the reverse part of that's going to happen. In other words, it's less likely that the enzyme is going to give up the substrate. You know, that the free enzyme, free substrate, and the ES complex that come into equilibrium with each other. But that equilibrium gets shifted when the inhibitor attaches to the enzyme. It kind of glues it all together and prevents the enzyme from releasing the substrate. And so that mimics high affinity. Does that make sense? I see about half of you saying yes, the rest of you giving me blank stickers. This does not make sense. Yeah, sure. So I have an enzyme. That enzyme binds to a substrate. Under normal circumstances, there would be an equilibrium between the amount of that ES complex that converts the substrate to product and the amount of enzyme that releases the substrate. Okay, because remember, the formation of the enzyme substrate complex is a two-way process, simply because the substrate just doesn't bond strongly to the enzyme. But when the inhibitor binds to the enzyme, it shifts that equilibrium so that the enzyme tends to hold on to the substrate better. It just doesn't convert it to product, because the enzyme substrate inhibitor complex is inactive. Was that clear? Does everybody get that? I'm crawling through this because I really want you guys to understand this. All right?
So when it is uncommitted, there's no product, when it's like that whole complex.
That's correct. That is correct. Which gets us to the second part of the question. What's happening to Vmax?
Vmax is increasing.
Is that correct? Yeah.
It's decreasing. It's less product is being formed.
That's correct. This is a funky one.
And going off of that is, it's because it stays locked in like the ESI complex, right?
Yeah.
When the inhibitors are there.
Yeah. So in this sense, it acts a lot like non-competitive inhibition in that it locks up the enzyme. All right, ready for an example? I got a fun example. Roundup. Y'all know what Roundup is? So Roundup is one of these famous weed killers that everybody sprays on whatever they want to kill it in their yard. And this is the technical name for it right here. And you can pronounce that if you want. N-phosphonomethylglycine or glyphosate. So, or glyphosate, whatever. Okay. Anyway, it is an uncompetitive inhibitor of 3-phosphoshikinate. Goodness gracious, that's a mouthful. 3-carboxyvinyl transferase. And so the way that they think that this works is that through this uncompetitive inhibition, it causes the buildup of a lot of metabolites, a lot of metabolic intermediates that aren't supposed to be there that become toxic to the plant. That's how they think it works. Okay, so they actually don't know exactly why this work or which metabolic substrates are necessarily becoming toxic. They just think that it just kind of shuts down some key metabolic processes, so you end up with an accumulation of a lot of intermediates. Okay, so that is an example of uncompetitive inhibition. Here's another fun example of competitive inhibition: lithium. So lithium is a drug that is used to treat certain types of depression. You know, a lot of issues that involve mood, depression, and one of the hypotheses, they actually don't know for sure how lithium works. They just know that in some cases it does. Not in all cases, but in some cases it does. And this is just one of the hypotheses as to how lithium works. What they think that it does is it interferes with the recycling of inositol right here. And inositol is part of a pathway for the production of gosh, these words are just really hard to say. Polyphosphoneinositide, PIP. That's how I call it all the time, by the way, is PIP, which is a key substance in a whole bunch of signaling reactions. Anyway, what it does, lithium, they think one of the things it might do is uncompetitively interfere with the function of this enzyme right here, inositol monophosphatase, IMPase right here, which regenerates inositol, which is then used to form PIP. And PIPs, they are second messengers in systems. A lot of them have to do with stabilizing mood, which is why, which is one of the things they're trying to prevent mood swings with lithium. So this is one of the hypotheses as to how it works. You know, if you were to go and say, for example, if you just went out and Googled lithium, you would find probably 3 or 4 different hypotheses as to how it's worked, but this is one of them. And if this is the way it works, it's an uncompetitive inhibitor. So lithium, and I should clarify what I said, lithium absolutely does this, but whether or not that drives the clinical result of lithium treatment is uncertain. Questions? So, got your brain spinning about competitive, non-competitive, and uncompetitive enzyme inhibition. Watch, had a lot of things going on with enzymes. Huh? Oh, we're not finished yet. Okay. Everything we've talked about to this point involves reversible inhibition. In time, the inhibition will go away once the inhibitors are cleared from the system. However, there are some types of inhibition that are irreversible. And irreversible inhibition, rather than involving weak molecular bonding forces that are very electrostatic in nature, they generally involve very stable covalent bonds, which means once the inhibitor sticks on, it is not coming off. Here is a really good example of this. You've all heard of nerve gas, right? Okay, a chemical weapon. Its chemical name is sarin. What sarin does is it is going to interfere with muscle function. So sarin interferes with the transmission of nerve signals to muscles. How does it do this? So we're going to get a little preview of some of our content on neurophysiology here. So nerve endings, they will stimulate muscles to contract. They do this when the nerve endings release a substance called acetylcholine. I'm sure you've all heard of that. It falls into a category of biological messengers that we call neurotransmitters, and we're going to learn a lot about neurotransmitters. And so this is a nerve ending right here. a nerve signal hits this nerve ending and it releases acetylcholine. What happens then is acetylcholine will bind to a receptor on the muscle cell. This will then open up sodium channels that allow sodium to kind of flow into the muscle cell, and through a lot of steps, this leads to muscle contraction. And when we get to neuromuscular junctions down the road, we'll talk about the details of how this works. The key thing here is that when the muscle contraction is done, there is an enzyme that hangs around the surface of the muscle cell. It's called acetylcholine esterase. And what this does is it will break down the acetylcholine. This is important because then that will close those sodium channels and turn off the muscle contraction. Without a functional acetylcholine esterase, muscles can't work. You'd open up those sodium channels, they'd be open up forever, and you'd be done. Okay? So what nerve gas does is it covalently binds to the active site of acetylcholine esterase. So this is nerve gas right here. This is acetylcholine esterase. It just pops itself right in there, binds to the active site, so it can no longer interact with acetylcholine. And this basically makes all your muscles non-functional. Very lethal. And this represents how a lot of poisons act. Another example of this is cyanide. So they have historically used cyanide in gas chambers to execute people. And what cyanide does is one little simple thing. It prevents oxygen from taking up electrons at the end of the electron transport chain. That simple little reaction. And it just totally clogs up the works. So you can no longer aerobically produce energy. Same deal. It is going to covalently bind to the enzymes that drive that and shut it all down. And endotherms cannot function anaerobically. Nasty stuff, yeah.
Would you suppose that you could maybe survive this with like something like an iron lung, where it's breathing?
Well, no, because you could have all the oxygen available that you want, but if you are enzymatically being prevented from that oxygen, which is what oxygen does, it's the whole reason you breathe oxygen is to pick up those electrons at the end of the electron transport chain, get them out of the way so the next pair can move through. Okay, and if oxygen is unable to do that, then having oxygen is going to do you no good whatsoever. Yeah, that's amazing. We're oxygen breathers. And the sole reason we breathe oxygen is to pick up those bloody electrons and create metabolic alarm. That's the sole reason. Seems like it's such a simple thing, but absolutely critical. All right, let's talk a little bit about how cells regulate enzymes. So cells have the capacity in some cases to produce substances that combine to enzymes and act sort of as on-off switches. So a substance can be produced that turns an enzyme off. We refer to this as negative allosterism. So when an allosteric regulator binds to an enzyme and changes its conformation so that the active site can no longer bind to a substrate, then that is how negative allosterism works. So an example of this here, phosphofructokinase. Notice I actually pronounced that word, PFK, it catalyzes the third step in glycolysis, where you convert fructose 6-phosphate to fructose 1,6-phosphate. And you can allosterically inhibit this just simply by having high levels of ATP. So the ATP in this case is going to serve as a negative allosteric molecule for phosphorofructokinase. Does that make sense? So I've got an enzyme that drives the third step in glycolysis. When ATP is high, it will serve as a negative allosteric inhibitor of the enzyme. Does that make sense? If it does, tell me why. What do high levels of ATP indicate? What is ATP? I know you all know this. Come on.
This can be clear off, ATP is like a source of like energy and like in the
Yes,
mitochondria.
it is the primary energy molecule in the cell. Remember, we talked about its key role in coupled reactions. It provides energy for all these synthetic things that we got going on in our cells to happen because it's the coupled reaction that makes the overall thing exergonic. Have a negative delta G. Okay. But in this case here, this particular enzyme, which is part of glycolysis, does it make sense that ATP would serve as an allosteric inhibitor of that enzyme? Yes.
Well, if you have high levels of ATP and it's not being used, like you don't want to just be wasting energy making more of it. And so you need glycolysis to happen for citric acid cycle and then phosphorylation. And so blocking it at the start.
Yeah, I mean, if you've got a lot of ATP, why waste energy making more? Because remember, it's not cheap stuff to make. You invest a lot of energy to construct that ATP because you need it for synthesis. It's the only way that you can overcome the laws of thermodynamics. You don't really overcome them, but you function in spite of them. Okay. I mean, we're all headed down the road of entropy. But this kind of, at least in a localized scenario, kind of defeats that. All right? And so, but you don't want to spend energy making ATP, and glycolysis is part of that process if you don't need it. So when ATP is high, it will inhibit the reaction. What does this sound like? Yeah. Negative feedback. Good job. I'd high five you if I was closer. High elbow you or something. There we go. Yeah, this is classic negative feedback. Okay. All right, that's negative allosterism. You can actually use allosteric molecules to turn enzymes on. There are some cases where you want to promote activity, and this is called positive allosterism. So, aspartase carbamylase catalyzes the first step in pyrimidine nucleide synthesis. Okay? In this case, ATP again is an allosteric activator. All right? And in this particular case, this was probably a little more challenging for you. In this particular case, it makes sense because you've got ATP, all right? You've got ATP because which is a purine. All right. And if you have a lot of this purine nucleotide that's present, and ATP is a nucleotide, in case you don't remember that. If you have a lot of this purine present, then it makes sense you want to balance that out with a pyrimidine. So remember, your nucleotides fall into two categories. You have purines, some are pyrimidines. And it doesn't matter whether we're talking about nucleic acids, RNA, whatever. Okay? And so this is kind of a way to balance out the production of those two categories of nucleotides. And so when you get a high level of purines, ATP will promote the synthesis of pyrimidines. What kind of feedback is this? Huh? I knew you were going to say that. Is that right? Is that really right? Are we maintaining homeostasis or not with this? What would be the homeostatic state in this case?
Would it be negative feedback since it's trying to balance out the protein?
Yeah, this is actually, that was a tricky question. I get it. Yeah, because the homeostatic state is a balance of the pyrimidines and purines. And so this is where you are promoting the formation of what you don't have in order to correct back to the balanced state, homeostasis. That was a little tricky. That's what you got to think about. When you're thinking about the difference between positive and negative feedback, the first question you ask yourself, are we maintaining homeostasis or are we not? If we're maintaining homeostasis, then I can't even think of an exception to that it would be negative feedback. Because negative feedback is always going to correct towards a set point. All right? Positive feedback is always going to correct away from a set point. And generally, it's going to amplify a response. Yeah.
So for positive and negative allosterism, is it always a set, like it's always giving negative feedback, or is it like?
No, I can't say that for certain. And I don't have an example of allosteric activation in my head that would be associated with positive feedback, but I would be shocked if there wasn't a lot of particularly positive allosterism. Well, actually, probably both positive and negative allosterism that would be associated with positive feedback, because you're having to amp up a whole lot of metabolic processes in order to get the response amplified. So, goodness, okay. I didn't even get through enzymes today, and I thought I was going to, but that's okay. Any questions? I will see you all on Monday. I've got another appointment at 2 with another group at 2 o'clock today, and then I've got an off-campus appointment at 2:45 today. So that's as late as I can go today. I've got my